cytoskeleton of a red blood cell. Red blood cells are weird, remember? They're atypical cells, they don't have a nucleus, they don't have mitochondria. We have this structure that underlies the plasma membrane in the red blood cell, and we can call it cytoskeleton. But in the red blood cell is very atypical. What do we see, however, for cytoskeleton in any eukaryotic cell? We see three kinds. And there are three kinds of cytoskeletal elements which are characterized by their typical, or their thickness of the elements. And so we have the thinnest element of the cytoskeleton, which is called the microfilaments. And they're about 8 nanometers thick, and the main subunit is actin. It turns out that actin in all cells is very dynamic, it's flexible. So these microfilaments form, break down, reform in the typical cell, not in muscular. Actin is important in muscle contraction, as we will see. Then there is the thickest part of the cytoskeleton, which is made up of the microtubules, about 25 nanometers in diameter. These are also pretty dynamic, meaning they form, they break down rather quickly, but not as dynamic as the microfilaments. And then there's a type of cytoskeleton that is in between in thickness, and therefore with a lot of imagination is called the intermediate filaments. The intermediate filaments are in between the thickness of microtubules and microfilaments. It's still pretty dynamic, but relative to the microtubules and microfilaments, intermediate filaments are overall pretty stable. Their structure is made up of a variety of molecules. Keratin is one type, and they're always very strong and rope-like. What I like about the cytoskeleton is you can really look at the relation between structure and function. We'll see. Overall, the cytoskeleton serves functions to support the cell, to give it some structure, and it allows various forms of motility. So what do we see if we look in some typical mammalian cell types, or what the cytoskeleton looks like? If we have in blue the microtubules in an epithelial cell, you see the microtubules run out from a central point. We see the intermediate filaments, they're pinkish, they tend to run across. And we see the actin cytoskeleton, the microfilaments, and we see a lot of it in the periphery. If we look at a cell in division, we see that the microtubules once again come from a central organizing point, the microtubules in blue. And we see that for the formation of the cleavage furrow, once again the actin organizes in the periphery of the cell. If we look at a neuron with a long axon sticking out, we see that the microtubules come from the cell body. They grow out from the central point and reach out to the periphery. And we see that the actin cytoskeleton, the microfilaments, are again more at the periphery of the cell. So we can kind of generalize that. And while it's not always true, it definitely serves a good purpose for your understanding. Most of the time it is like this. So microfilaments are made up of actin. We'll see that later. It kind of looks like beads of actin string together. We'll see that in a couple of moments. If we look at a fluorescent micrograph of a typical eukaryotic animal cell, you see a lot of the microfilaments are in the periphery underlying the cell surface. For the microtubules, which are hollow tubes, you see that the microtubules tend to run out from a central point to the periphery. I think it's visible in this fluorescence image. The microfilaments, it's harder to say, but they tend to run across a cell. But we also know that there are microfilaments that are around the nucleus, or actually within the nuclear envelope. They kind of surround the nucleus. So we can generalize, and we can say the microfilaments tend to be in the periphery. And we also know that the microfilaments grow in a certain direction, and they tend to grow fast in one direction, not so much in the other. That's what we can indicate with an arrow here. The microtubules, we know, grow from a central point outward. And so the typical organization is outwards. Whereas for intermediate filaments, the typical organization is surrounding the nucleus and then across the cell. And I hope one thing that we're going to understand before and after the spring break is that these organizations and the structures that we see here for the filaments actually make sense for the functions of these different parts of the cytoskeleton. So I really like you to understand this diagram. Knowing that it's a little bit of generalization, that's overall pretty true. It's just that in plant cells, the microtubules can have a little bit of a different organization. Okay, so keep in mind, the microfilament is very dynamic in most cells. Microtubule is still dynamic, intermediate filaments the least dynamic, meaning the structures are more stable. Okay, let's look at the microfilaments first. Microfilaments are in all eukaryotes. What's their function? They support the cell, they support the plasma membrane, they give the cell the shape, therefore. They're allowed in motion of an entire cell, and in animals, they're part of the mechanism for muscle contraction. Microfilaments are always made up of actin. Actin is a protein, and it's immensely conserved, meaning actin in us and in yeast is virtually the same protein. Very conserved protein. It's also quite abundant in our cells. It's one of the most abundant proteins in most eukaryotic cells. Actin binds ATP and it can hydrolyze that ATP, which typically happens after the incorporation of the actin in the filament. The monomeric actin protein, actin is a protein, the monomeric actin protein with that ATP bound is globular, that means it's like a soccer ball shape, globular, and we call that the G-actin. There always is a pool of some G-actin in the cell. Now, if actin becomes incorporated in a microfilament, then we speak of F-actin, filamentous actin. So that globular actin, that's just the monomer, filamentous actin, that's many actin put together in a filament. And here we have a picture, shadowing picture of an actin filament. And based on the structure of actin monomers and these types of pictures, we can come to a model where the monomeric actins have all been colored in the colors of the American flag, which is also the colors of the French flag, the Dutch flag, the flag of Luxembourg, and Russia, and many others. But let's say today it's the American flag. So here, in white you have one monomer, in red another, and in blue the following white, red. It's just to indicate what the different proteins are. Yeah, red, white, and blue are all the same protein, right? Now we can see that there's actually space here where the ATP can bind. So we know this is the ATP binding site, and we call, just by definition, that end of the actin monomer the minus end, and that would make the other end, if I'm pointing at this one here, this is the minus end, and the plus end is the other side. And we call it minus and plus because if you look at the filament, it turns out that the plus end is what grows the fastest. If that filament forms, because the filament forms by actin monomers coming together. Okey-doke. So, how do we know about the growth of these actin filaments? It can be studied in a test tube. I told you, actin is a very abundant molecule in eukaryotic cells. It's not difficult to purify it, and people have done that. And you can put that actin in a test tube, and you can see if you can grow filaments. So what do we see? It turns out that you just need a lot of actin together. And an actin needs to be in the ATP bound form. It's a minimum concentration required before you ever see the growth of the filament. We call that critical concentration. Once you have that, you will get the spontaneous growth of a filament, that 8 nanometer thick fibrous shape. And if we look at how that grows, we see that there's one end of the filament that grows quickly, and that's the plus end, and the other end grows very slowly, and we call that the minus end. Now we know that if actin is incorporated into the filament, and we see hydrolysis of the ATP, and the ADP bound actin stays in the filament, but filaments made up of ADP actin are a little bit less stable. So the ATP hydrolysis, which will happen, destabilizes the filament. Not all the actin filaments in a cell fall apart all the time, and that's because there are proteins that bind to the ends of the filament, called capping proteins, and they stop the growth, but they also stop the breakdown. Now, what we can see, if there's growth fast at the plus end, but actually if there's degradation of the filament at the negative end, which can happen when concentrations drop, then in a test tube, we actually see the actin filament grow on one end and break on the other, and it's kind of like track milling. Now, in the cell, the idea is that there are many proteins that regulate how the actin assembles and disassembles. You can look at what happens in a test tube. So if we have a lot of actin in the ADP form, we just get spontaneous formation of filaments, and you typically get a plus end fast growth and slow end at the minus, so slow growth at the minus end. If the concentration of free actin drops because it becomes more and more incorporated into filaments, you see that the growth at the negative end stops, and actually in the next reaction, subunits drop off, whereas the addition at the plus end keeps going. So over time, these subunits here, which are indicated in the lighter color, are removed, whereas more is added to the plus end. That's a track milling effect that we can see in vitro. Okay. Now, in the cell, how an actin filament grows is going to be controlled by proteins that bind here and there at the plus and minus end, and by proteins that control ATP incorporation into monomeric actin. So in a cell, there's a lot that happens to control how the actin cytoskeleton forms. Here we have monomers polymerizing, forming these filaments. The filaments can be cross-linked. There are proteins for that make networks. There are proteins that bind to the ends, that's capping, and that stops growth but also breakdown. There are ways in which actin can be bundled together to organize it. There are proteins that can promote the disassembly, and a protein that's well studied for that purpose is cofilin. It promotes the disassembly of filaments. There are also proteins that promote the assembly. Profilin is a protein that promotes the assembly because it reacts or binds to actin that has ADP bonds. It kicks out the ADP, and then because there's a high ATP concentration in the cell, that ATP comes in. So profilin is an exchange factor. It exchanges ADP, letting ATP in. And if there's high profilin activity, you promote the actin to be in an ATP state, which is competent for assembly. So you promote filament growth. And then there is a way in which you can keep monomeric actin from incorporating into filaments, and that's achieved by the phimosin protein. So you can imagine if there's a cell And if it wants to disassemble actin at one point and assemble it at another point, then it might want to have the phimosin proteins and the profilin proteins at the point where the disassembly needs to be favored, and the profilin at the site where the assembly needs to be favored. And that's what's going on. So actin serves a lot of roles, but one role is the motility of cells. And you can see this, for instance, for the growth of a neuron. We have a neuron here. This is a time-lapse, light microscopy picture. It's just phase contrast, and we see here a neuron. Shoot a picture a little bit later, it has grown. That growth of that axon is due to actin. And here we can see the tip of such an axon. And this is just a phase contrast image again. If we do staining using fluorescent microscopy, we can see here in green the actin. And the actin is polymerizing and it's pushing these small feed-like elements out from the cell so that it can grow in this one direction. Or if you take a connective tissue cell, such as the fibroblast, it can crawl towards a site where it's needed. For instance, in wounding, fibroblasts need to be part of the repair system because they make connective tissue. So they fill up a wounding situation. In the case of a wound, they fill up the space. So fibroblasts can crawl, and we can see in this fibroblast, here there's actin polymerizing at the leading edge where this fibroblast is growing towards. So the proteins that promote this assembly of actin are here, and the ones that promote this assembly are at the tail end of this fibroblast. And that's part of its crawling mechanism. Now some bacteria, I'm going to end here, some bacteria have learned to misuse this, and Listeria is one of them. And we can see here a Listeria bacterium, and it's actually recruiting the actin to polymerize behind it, making for a tail of actin that pushes this bacteria through the cell. And here we can see the bacteria in yellow and the actin in green. And you can see the Listeria moving through the cell, and it can even get itself pushed through the cell membrane into the neighboring cell. So there are some pathogenic bacteria that misuse the actin cytoskeleton. They misdirect the assembly. I'm going to keep it at that.